Hello and welcome to the Algorithms and Data Structures for Bioinformatics course. In this first lecture, I'm going to give a brief introduction to the course, explaining a bit the motivation and the context in the basic notion of bioinformatics, talk a bit about how what we're going to do here in this course fits with the science of bioinformatics and life sciences in general, and also explain a bit how we're going to structure the course and how assessment will be at the end, so what will affect your grades at the end of the course. But first we're going to start with bioinformatics. This word has been used in many different ways, and actually part of the thing it refers to, which is the use of computers to help with molecular biology problems, even predates the word. So around the late 1960s, computers began to be used especially to help process protein sequences because due to the developments in Edman degradation and sequencing proteins, a lot of information was beginning to be gathered, and so computers were used to process this information, try to find patterns, similarities in the sequences, and so on. And also in parallel, there was growing in the biological community this idea that these complex molecules like proteins and DNA would carry biological information. So in the 1970s, theoretical biologists Hesper and Paulien Hogeweg proposed this term bioinformatics as referring to the discipline that studies information flows in biotic systems. So the idea is a bit like we have biochemistry, which studies the chemical aspects of biological processes. Biophysics studies the physical aspects, and bioinformatics would focus on the information aspects of biology. And we can see information in the way, for example, that evolution will, due to selection pressures, mold different features in organisms and in molecules and select for different attributes. So we can think of that as evolution informing the molecules themselves and the organisms. We also see information flow, for example, from DNA to proteins, proteins through their interactions to the development of organisms, and then eventually to interaction with the environment and evolution. And we also see in many biological systems information flow in molecular signaling, in the protein modification pathways, enzymes, and so forth. So the idea here would be that bioinformatics would study these systems from the perspective of looking at how information flows. Nowadays, the term bioinformatics has been used in many different ways, and we can consider it in a broad sense to be generically the application of computers, of computer science to biological problems. So we have problems like data analysis, storage and processing, especially managing large databases of information. We have things like simulations and numerical methods for modeling processes. We have machine learning, representation in things like ontologies and so on, the use of artificial intelligence. There are lots of different types of approaches, but they all fall in this broad sense of bioinformatics as meaning using computers and computer science to help solve biological problems. There is also sometimes the use of the term computational biology, and some people try to make these terms mean different things, or sometimes they mean the same. For example, if you go to the subject on Nature or on the medical subject headings, taxonomy, you'll see that bioinformatics and computational biology are synonymous. And it's mostly viewed as this interdisciplinary field that involves computers and computer science, but also biology, molecular biology, and so forth. So we are not going to worry too much about this, about how exactly people use these terms, because this varies a lot in different contexts and along the time, because sometimes these words are used as buzzwords, and to keep them fresh, people start using them in different meanings. But for us, the idea here is not to make these kinds of distinctions, whether bioinformatics works on sequence data alone or whether it refers to software development or the application of software or something like that, but rather looking at it more like a discipline on its own. So if you want, you can consider that it's interdisciplinary in the sense that you have background knowledge from computer science in software engineering, database management, AI, and so forth, but also from biology, because you need to understand the problems, genetics, proteomics, all the structural bioinformatics parts. You need to understand the underlying biology and biochemistry. But we can think that this is actually a discipline on its own, because the interdisciplinary nature is just a result of how we split apart different aspects of science and then bring them all together again in bioinformatics. So I think the way we are going to consider the field that we'll be working on is more in this idea of bioinformatics as the discipline that focus on the informational aspects of biological systems. So if we are aligning sequences, we are trying to find how information flowed during evolution to make some parts of one sequence homologous to other parts, and then we align them and try to match them together. We can also use that information to infer phylogenetic trees, try to figure out how the sequence of a protein, for example, informs its structure, and so on. So this would be the basic idea and our motivation for looking at algorithms and data structures which underlie the implementational aspects of this approach. Another way of looking at this is in the same way that we have information flowing in biological systems, so DNA to messenger RNA, protein, then protein structure determines function. Function will determine the characteristics of the organism, and this will in turn interact with the environment and lead to selection pressures and evolution and so on. We also have something similar, some information flow in the actual process of trying to understand these things. So we can think of a caricature or an outline of scientific reasoning as starting with some hypotheses, we experiment, and then we evaluate back to the beginning and try to generalize and so on. So we are working with information flowing in this process too. And in bioinformatics, the two things come together inevitably, because we are not just trying to understand and model how information flows in nature by itself, but also as part of scientific research in order to understand how nature works. So in this context, we have theoretical aspects of bioinformatics. We have methods, algorithms, generic approaches like machine learning and so on. We have specific applications. We may want to assemble a complete genome from fragments of the sequence, or try to predict protein structure, organize databases, and so forth. And we have the data itself. We have lots of data that we obtain from laboratory experiments, from cross-referencing information. So we can actually use bioinformatics to generate data from the more elementary parts. And so we have a large amount of data. And this will give us, so to speak, the landscape, the context for bioinformatics. We have a huge amount of data and very complex systems, and that is what we're going to try to manage using the computer to help us. When we are trying to understand some aspect of nature, we can think of the process of scientific research as focusing on three different parts, three different aspects. We have that part of nature that we want to understand, some specific process or the role played by some kind of molecule or something like that. And then we have the process that we are using to try to obtain knowledge from the examination of those processes, the data that we have. And as part of this, we have the computational aspects, where we use computers to gather that knowledge and to apply that scientific knowledge. But the important thing here is that there may be, and there often are, differences between the scientific theory that we are creating and how we can put it, so to speak, in the computer in order to use it and to compute with it. So let's think of the science itself. We have different things like the models that we create to represent some aspect of reality. So we can have equations, diagrams, we can have descriptions, and then we have hypotheses about how well these models correspond to some specific aspects of nature. For example, we can have a model of the Earth as being a perfect sphere, and this will not match perfectly with the Earth, but it will be a much better model than if we consider the Earth to be flat or a cube or something like that. So those specific things where we instantiate, we have specific parameters, something like that, we can obtain hypotheses that connect our models to nature. And then we have this theory that gives us a framework for how to build models. So this is something that is true in general for science. It also applies to the parts of science that we try to put into the computer for computation. So our computer models that we create are also attempts at representing some aspect of reality. We must also take care to validate the hypothesis that these models actually correspond to what we want to describe when we test them. And there are theories behind it. So there is a theoretical framework for building these models, for understanding what we can actually put the computer to work with, how we can do it, and all the theoretical framework also that is relevant to the type of problems that we are trying to answer. However, the computer model, even though it's also a scientific model, it must be specified for the computer. So we cannot use the same kind of language that we can use when we are explaining or transmitting a model to other humans. And also we must bear in mind that the purpose of codifying the model so that it's specified for the computer is also so that the computer can do the computations and give us the results. So this adds a constraint to the kind of models that we can work with and create on the computer. It's not any scientific model, but it must follow those constraints. I'll give you an example. We can suppose we want to find these contexts, the constants, the maximum velocity and the Michaelis-Menten constant for an enzyme, and the enzyme follows this classical Michaelis-Menten kinetics, where the velocity of the reaction will depend on the concentration of the enzyme, the concentration of the substrate, and these two constants. So the catalysis constant gives us the maximum possible velocity, and this Km gives us the concentration of substrate at which the velocity will be 1/2 of the maximum. So this would be our scientific model. We can specify with these equations how a description of this aspect of reality, how the enzyme works. And now we want to create a computational model that allows us to find these parameters from a set of experiments. So we can gather the velocity, we can measure the velocity of the reaction at different substrate concentrations, and then use those values to compute the constants of the maximum velocity and the Km. So this would be an example. We have our different experiments here on these crosses. Let's suppose that this blue line is the actual curve. So this would be the real curve if we knew the actual values. But since our experiments always have some error, some noise, we are computing a different curve. In addition, not only because our experiments have some noise, but because there is a difference between the scientific model and reality, and a difference between the computational model and the scientific model. Because in order to find these parameters, we must do something, implement some kind of algorithm. For example, iteratively reduce the quadratic error between the curve that we are computing and the data points that we have. If we do this many times, so iteratively we are reducing the error, but at some point we stop, and there will be still a computational error left over because we are using an approximation to try to find these values. So not only we have the problem that data is noisy, so every time we try to gather data, we have that problem. We also have a problem that scientific models may not correspond exactly to reality, but rather they give us an approximation in some conditions or under some assumptions that simplify the model. But then when we try to put these scientific models into the computer so that the computer can tell us some result, we have to bear in mind that the computational model itself will not necessarily correspond exactly to the scientific model. We may have to do some further approximations or further assumptions in order that the model is computable. So we are, in bioinformatics, we are trying to use computers to implement and run our scientific models, but we often need to approximate the scientific models in order for them to be computable. There are two things here that we need to bear in mind. On one hand, we need to specify the models in some kind of language, in some way that the computer will be able to operate on. Talking to a computer is not the same thing as talking to a person, so we cannot use the same kind of languages and metaphors and ideas that we use in science when we are creating computational models. Furthermore, since we want the computer to run and to give us a result, the way that we implement these models must allow the computer to run in a feasible time so that we can have the result in a useful amount of time with an adequate amount of resources. So if the computer takes 3 million years to give us the result, that's not useful. We don't want that kind of model. However, for this we have a wide range of applications. There are many different techniques that we use in bioinformatics, and it's a very broad field. So in bioinformatics, our focus on this particular course will be on how to use the computers. So we are going to focus on algorithms, the way we specify all the different steps in order to run the computations that we need for our models, and also data structures, which is how we're going to organize the data in order to process it. So for this course, in a quick outline, we will take into account that although bioinformatics is a rapidly changing field, you have constant innovation not only on the experimental part, on molecular biology and so on, but also on computational techniques, on hardware, and that is leading to huge changes very rapidly. However, the basics of the approach is stable. So the basic ideas of how to reason computationally, how to implement algorithms, how to think like the computer so that we can tell the computer what to do, those are very stable at the core of bioinformatics, and that will be our focus. So we are going to look at some different problems and different algorithms. We're going to sample them, choosing in preference simpler examples of those algorithms so that we can look at them in detail, and then you're going to see how you can implement them. So we're going to use Python for this, and we are going to look at these algorithms in sufficient detail so that you have an idea of how you need to structure the data, how you can specify the different steps, and so on. To study for this course, you'll have the slides, lecture notes, and the recordings of the lectures on the website for the course. And also, I recommend at least these 3 textbooks because I will usually use them as the basis for many of the lectures. There are some other papers and textbooks that I may refer, which you optionally can consult, but these would be the core textbooks that I would recommend. For the class plan, you will have these 2 lectures for each week pre-recorded, so I'll try to make them available 1 or 2 days before the schedule, and I will try to make them in total no longer than 90 minutes. So the idea is that if you want, you can view them before the scheduled time for the lectures, or you can view them at the scheduled time and still be in time for the discussion session. The discussion session will be the last part of that 2-hour block for the lectures, so from 2:30 p.m. to 3 p.m., where you will have a Zoom session for questions, for discussion, where we can talk a bit about the subject matter. Although the lectures will be recorded, so they will be available even after the scheduled session, I recommend that you view the lectures before the Zoom session so that we can clarify any questions that you have. The tutorial classes will start on the 6th of October, so on the 2nd week, and they will also be online, but we were going to use Google Hangout. So most of the questions that you may have in the exercises will probably I'll be able to answer them via chat, and the historical will remain on the Hangout, so you can read up on previous questions or review them if you want for the assignments or the tests as needed. But also in the case that you have some specific issues with your code and we need to share something, we can also do some video calls as needed. So I will post on Clip or send you by email these kinds of links so that they are not public in the website, but that you can access them in any way. So we're going to start with this first introductory stage, the first few weeks, where I'm going to talk in general about algorithms, about some techniques like dynamic programming in next week. And then we're going to start looking at sequence alignment, but still without writing code. So you're not going to start programming because I do not assume that you have previous knowledge or previous experience with programming. However, in parallel with this course, you are also taking an introductory course in Python. So after a few weeks, we'll be able to start taking the first steps with Python and start building on those in order to reach the same kind of complexity of the algorithms we'll be looking at. So we are going to start with sequence alignment at a higher level. But at the implementation level, we're going to start with simpler things like identifying regions in subsequences in DNA, for example, things like that, so that you can start becoming more comfortable with Python before we move on to more complex algorithms. Then we're going to look at sequence search algorithms and multiple sequence alignment, phylogenetics, and at the end of the semester, we will move to machine learning algorithms, which are a bit different. We are not going to look into so much detail at the implementation, but a bit more on how to use them in some cases, and a bit on structural bioinformatics at the end. So note that this is the first edition of the course, so this is the plan at the moment, but expect some changes and updates to the planned classes. But in any case, I will keep everything posted on the website, and you can keep track of things there. For your assessment, your grade will be split into 2 parts. The theoretical part will be either with 2 written tests during classes or an exam that will be taken after the classes end, so I think in January. I'm not sure if these will be online or in person. It depends on the rules that will be in place by the time we take the tests, but we'll see how we can do them. In any case, the exam will be scored in 2 different parts, each part corresponding to 1 test. So if you take the tests, you have the grade for each test guaranteed, and then if the exam doesn't go so well, you at least have the grade from the corresponding test. The other part of the assessment is practical, which is going to be split into 2 assignments. I'm not sure yet if the assignments will be related or completely independent. Probably they will be independent, but I'm still thinking about that. But the assignments will be where you write a short report, a bit of code, and do some more hands-on work, not the theoretical part like in the tests. The average of these 2 components will be your final grade. So to sum up for this introduction, we saw a bit the idea of bioinformatics, or different notions of bioinformatics that we have, how this fits into the scientific research in biology, and this idea of bioinformatics as the study of information flows in biology and the use of computers to help us study those aspects leads us to an important notion that we are in bioinformatics writing or specifying scientific models in a way that the computer can understand. This leads us to 2 important constraints. First, the kind of language that we can use and the details that we need to specify. And the other is that we want to obtain some results. So this specification must lead to some kind of procedure that the computer can actually follow in a practical amount of time with a practical amount of resources. If you want to read more about this part, I strongly recommend this paper by Paulien Hogeweg, which is on the fundamental ideas and the roots of the term bioinformatic in theoretical biology, and then the first chapter of this Burkowski structural bioinformatics. This is optional, it's not very important, but it talks a bit about some of those aspects that I mentioned. The first chapter of the Bioinformatics Algorithms book is also a good idea to take a look, because it gives you a roadmap of the kinds of things that we're going to look at, implementation and so on. And if you don't have a good background in molecular biology, I would advise you to read chapters 1 and 2 of the Understanding Bioinformatics book. So this is just background on DNA, proteins, and so forth. That is useful if you don't have that background yourself, because in most of the problems that we're going to cover, I will simply assume that this is information that you already have, because most people in this course come from life sciences background. So if you are in some doubts about that, take a look at these first 2 chapters so that at least you understand the basics for these problems. Okay, so that's it for this first lecture. Welcome everybody to this course. And you also have available by this time the second lecture. Thank you for your attention.